So here, what we're dealing with is the first of your FRQs, and it says that the sex of an organism is typically determined genetically, but that sometimes you can have environmental factors that influence that sex. So looked at genes and environmental impact and sex determination in individuals with ZW, pair of chromosomes, ZW, Z and W, so one Z and one W, were always female. So ZW, they put F for female. Where individuals with ZZ were either male or female. Depending on the environment. Now, they did a weird notation that you may not have been familiar with when, honestly, I wasn't with either in the beginning. But ZZ male and ZZ female. And they just made this female just to remind you that it's always female. That's all they did there. Annotating helped me a lot. I'm sorry, I have to pause here to ask people to be quiet because even though I'm wearing noise canceling headsets and you're supposed to be working on your FRQs, I have 3 people talking to each other. I have others just having very broad conversations, so I have to pause here and figure out what's going on. So we have scientists examine the roles of genes in environmental temperature and sex determination, right, in lizards. And ZW means that you're female always, and they put a little F there just to let you know. And ZZ is either male or female, depending on the environment. Scientists made it one ZW, which is always female, with a ZZF females, or ZZM males, right? So they took these and mixed them and crossed them. So ZZ male with a ZZ female, and ZW female with a ZZ, one of these two with a ZZ male, and incubated the eggs and produced 20 clutches at temperatures ranging from 23 to 36 degrees Celsius. There's a temperature. And when they hatched, the scientists determined the sexes. And then they added an interesting note that those that did not hatch, they were approximately the same for males and females. So that was not a factor. Now they have some interesting notations here. And so we should look at that really quickly. They have that plus or minus. And this is a plus or minus standard error. Here's a plus or minus standard error of 2. So whatever number you get, on average, the mean standard error is plus or minus 2, where each of the individual measurements, all right, is plus or minus 0.08. So we looked at 23 degrees, you had a 50-50. These are decimals, but I hope that you know that these decimals mean percentages. It's a fraction of female progeny. So that's 50% plus or minus 8%. So it could be is 50 minus 8% or 50 plus 8%. So it could be up to 58% total. So plus or minus again is your error, the amount of error in the measuring or counting. And here they're all 50% until you start to get here. Now notice that the percent error goes down as well as you go up in temperature. And you see here that 0.58, 0.74. Here's at 36 degrees, it's 100% with no error, no standard error at all. 100% are female. Where here 50% are female. Now, when you cross a ZZ male, which is can be male or female, in this case male, with a ZZ female, so ZZ female, right? You have 0% plus or minus plus 2, 0% female until you get to 30 degrees, and you end up with at 100% female at 36 degrees. So this first question here, A, asks if you had a gene on the Z chromosome, and you had a ZW genotype, why do you have a greater probability of expressing a recessive phenotype than someone who has a ZZ genotype? Now, this is actually analogous to the XY-XX inheritance pattern that you learned about in ninth grade when we did pedigrees. So, when you have a ZW, if this chromosome has on that chromosome, the Z chromosome, you have a little a, and on this W chromosome, you don't have the A gene at all, because that's the whole point of having a different chromosome, have different genes. Then you only need one little a to show the recessive phenotype. Where if you had a ZZ, you'd need two little a's to show the recessive phenotype. Now, in ninth grade and in previous work, you would have noted that it's it's a very same situation with X-linked traits. So if on the X chromosome you have a gene that has a recessive, let's say big R or little r, and if you have two X's with two little r's, you have some, let's say, dwarfism. This one form of dwarfism is X-linked. Now remember, the second gene comes from the other parent, right? So if this parent gives a little r to this child, then this parent must have given, this is the female, this is the humans now, and this is the male. Then a male has an X and a Y. The Y doesn't have the R gene, that's a different chromosome. So this has the R gene. If that male, if that male has a little r, then there's a good chance that will give the little r then this person will definitely be a dwarf, because this mother, this parent, has two little R's, then this child's definitely going to have a little R. However, if the father's not a dwarf, if the father has not a dwarf, then there's no way for the female to be a dwarf, because the female can get sorry, I'm trying to erase this. The female is going to definitely get a big R from dad. Now, if that same chromosome was given to another egg, so you have the little R, but dad gave a Y, so it's a boy, then this male child, will definitely be a dwarf, even though it only has one little R. Where this female has a little R, but it has the dominant version, the non-dwarf version of the gene. So it turns out that with recessive traits, the female on the X chromosome, X-linked genes, recessive traits are protected in females. You're protected from recessive traits because you have two chances to get the dominant allele. Where in males, you only, on X-linked genes only, you only have one, there's only one chance to get the dominant allele. So you have, and that's what they try to discuss with you here with the ZW. If you have a ZW and the gene that we're worried about is here on the Z chromosome, so it's only found on the Z chromosome, then if you get one recessive allele, then you have the recessive phenotype. Where if you had ZZ, you'd have two chances to get you'd have two chances to get the dominant phenotype, so that you wouldn't have recessive here. But why didn't you get recessive? Because you had that second opportunity. This is all remembering that you have two parents. Each parent is contributing to the zygote and the sperm or the egg. So if you have someone with a ZZ, in this case a lizard, in the case that we just talked about with humans, X-linked traits, having two chromosomes gives you doubles your chances of getting the dominant phenotype. So the answer to this question, why do they have a greater probability of expressing? Why does someone with a ZW have a greater probability of expressing the recessive phenotype? Because you only have one copy of the gene, where the ZZ has two copies of the gene, giving you twice as much, reducing your odds of getting the recessive phenotype by half. And the person that answered this did a good job. It would be marked as correct. Using the template to construct an approximately labeled graph, including error bars that represent the table 1, ZZM, ZZWs, determine whether there is a significant difference between two crosses in a fraction of the female population. All right, at 24 degrees and at 36 degrees. So your objective is to identify the 24 degree difference, 32 degree and 36 degrees. And they want you to a labeled, select an appropriately labeled graph, including error bars for table 1. Well, if you look at table 1, the first question is what am I going to put on the x, what am I going to put on the y? The x is going to be the independent variable. Which of these is independent? Well, the temperature is independent because you control that. And the percentage of female progeny changes with the temperature, not the inverse. So here I would put temperature. Let me zoom in. Degrees Celsius. Then I would put fraction of female progeny. It's quickly sketching it because you're not going to have a lot of time on a test, on an AP exam. And it says, I look at the scale and I look at 0.5 and it goes all the way to 1 for the y-axis. What I like to do is I don't like to start at 0. I don't like to start at 0.5 because I like to give myself some room to predict below or above the minimum and the maximum. If I have, let me move this over. If I have 50% and is my lowest number and my highest number is 100, I can't go above 100%. And I'm going to put frequency of female over here. Fraction, I believe it's not frequency, fraction. But let me make sure that's clear because if you're doing it on the AP exam, it's important. But I'm running out of time, so I'm just going to go a fraction of females. Plus or minus 2 standard errors. So if I started to, that's the title. These are the two titles. That's what's on the y and that's on the x. That's what they're going to grade you on, whether you know what your independent dependent variable is. The next they're going to ask, do you know how to set up the graph? And so the setup should be, you have to have your minimums and your maximums, but you don't want your very lowest one to be the lowest one you have on your graph because then you can't predict below your minimum or above your maximum. So let's go ahead and start with 0 here at the beginning. And if this, we're going to set this as 100 because you can't go above 100%. So, you can't go over the max that you have. You can't create new eggs out of nothing. So from 0 to 100 is fine as far as the scale is concerned. If you count, it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. There's 20 divisions on the y. 20 divisions on the y. So each one of these is going to be worth half of 1, right? Or 5% each. 5% each. So 5, this is 10%. This is 20%. This is 30%. This is 40%. This is 50%. This is 60%. This is 70%. This is 80%. It's 90%, and of course that's 100. Now you might be able to do a little better. You might want to bring it down. You might want to, bring this 50% down here. But the problem is you have zeros here, and because you have zeros here, you're going to have to keep it something like this, where you start with 0 and you go all the way up to 100. 0 and the highest is 100. because both these lines, this is going to be one line, this is the other line. So then you're starting at 23, you're ending at 36, and you got 20 divisions going across, right? So the difference between, so what if I started at 20 instead of 23 and ended at 40 instead of 36? That gives me 20, so I can easily draw it out. So my x-axis, I can start at 20, and just then each line is worth 1. 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, and then, of course, 40. Now, there's no 40 on here. There's no 40 temperature, but now I can predict what would happen at around above 36, which is 36 is all I measured, is as high as I measured. But if I do a trend line, I'll be able to predict what would happen at 40 or 37, even though I don't have the data for it. And I can go below 23, which is the first data point I have, and go all the way down to 20. And if I expand the graph, I can go all the way down to 0 and all the way up to whatever temperature I want as well. So that's the power of a graph and the scaling of it. So then I would go ahead and plot my points. So I know I'm looking for 23, and I know 23 is going to be 21, 2, 3. So 23 is here, and that's going to be 50%. So 23 and then 50. So I'm going to use different color because I can, I'll use different colors, but obviously you're not going to be able to on an AP exam if they ask you to graph. So at 23, 1, 2, 3, I'm going to put a triangle, as I'm going to use the triangles for ZW. The yellow triangles in this case are going to be ZZ males crossed with ZW females. All right. And so that's my triangle. My first triangle I have is there at 50%. And then at 24, it's still at 50%. At 26, it's at 50%. At 28, it's at 50%. At 30, it's at 50%. At 33, it's at 58%. 58, that's 55, 6, 7, 8 should be about here. Obviously, you're going to have to do some guesswork, some estimation. At 32, I'm sorry, I'm already at 33. At 34, it's 74%. At 35, it's at 93%. And at 36, it's 100. All right, so if I were to draw the best fit line, it wouldn't be a straight line. It would be something like this, kind of a exponential curve, logarithmic growth, and then we have exponential growth there with the fractions increasing quickly after somewhere around 30-something, 32. After about somewhere around after 32, we have a real steep increase in the number fraction of females when I cross ZZ males with ZW females. Now I'm going to go ahead and do the same thing with the other graph, ZZ males and ZZ. So I'm going to have a different color, obviously. That's pink. Let's choose pink, and I'm going to use a circle instead of a triangle. And that's ZZ males crossed with ZZ females. So when I do that, I can start at 23. Again, starting at 23, I have 0 that are female at 23. So 1, 2, 3, 0 are female for this particular cross. And next is 24, and still 0. 24 is still 0. And 26 is still 0. And 28 still 0. And 30 is 0.03. So if 2 lines is 10, then I have to guess it somewhere in between. 3 would be somewhere around here. That would be about where 3 would be. Again, there's some estimation going on because our numbers, lines are not fine enough, refined enough to be able to fit everything in. So that would be at 20. Oh wait, did I mess that up? At 28, it's still 0. I don't even know what I was reading. Sorry about that. At 28, it's still 0. Still 0. At 30, it becomes 0.3. At 32, it's down to 0.2. So it's kind of flip-flopping. Since it's plus or minus, you see how it says plus or minus 0.01, really, these two are probably the same. And here is at 33, it jumps to 0.5. Oops. Misread that again. I apologize. It's not 0.02. This was 0.03. That 30 was correct. It was correct. It is 30. Here is 0.02, which is 2%. But then, see, 0.03, which is 3%. I'm sorry. But this is actually 20%. So I have to jump up at 32. It comes up to 30%. So it's all the way up here. And then 33, it's 0.52. So 33 is 0.52. Just somewhere around here. And then 34 is 0.88. Something like that. And then 35 is 0.97. It's going to have to be a little bit of an overlap. And then 36 is 1. So they're right on top of each other. So again, if I would have drawn the line here, let me draw this line in I don't know what color to use. Orange, yes. You see there's a best fit line that we kind of curve up, a real steep curve here, and then it goes here. So those are the 2 lines I would draw. And then, of course, our standard deviations our error bars are going to be, I really hate that they ask you to do something so detailed as this. This is absolutely ludicrous. But for 23 here, you see that 23 is about 8% plus or minus 8%. So I have to actually zoom in to do 23 for the first one. I'm working on the yellow triangles first. So it's 0.8%, right? So that's plus 8 and minus 8. So it would be 58 at the highest end. Go ahead and use, I don't know even what color to use anymore. Let's use purple for error bars. Give me a second. So 58 would be the highest. So if that's 50, that's 60. So 58 would be around here, from here to here. That would be the high end, and then underneath it, 8 down would be somewhere around here. All right. And then if I did 24 is only 2% off. So 24 would be at 2%. If there's a whole 10 there, that's 5. So 2 would be here to the center point, and then 2 down would be here to the center point. So the error bars are smaller there. And we got, what is the next one? The next one is at 26, and that 26 for the triangles is going to be 0.07. So 5 would be here, 6, 7. A little smaller than 8. Not quite as far as 8. Again, there's some estimation involved. Then 28, you have 3% off. At 28 is 3% off. So at 28, we're looking at something like that again. It's a little more. This probably should be a little more than 0.2, but my guess is that they would be okay with that. And 30 would be 0.1. So it's only 1% off, very close, almost no bar at all, really almost no error on 30. 32, 3. There should be a triangle there. I don't know if I messed up or not. 32 should be at 50% here. And it should be 3. It should be about there. And then that's 32. 33 should be at 58. It is. And it should be plus or minus 3, so something like that. Hard to read, actually. And then 34 should be 4%. And then 35 should be 3. And then 36 is no error at all. So it's hard to draw here. I would imagine it would be difficult to do on a test as well. It's really annoying that they have you draw error bars on it. They do ask you to do it, though, don't they? They say very clearly, appropriately labeled graph. That I'm assuming they give you to have more graph paper, more space than I've given myself here, so that would make life a little easier. You would put the title fraction of females versus versus temperature in degrees Celsius would be the title. You'd want this posted on your graph somewhere or underneath it or around it, so that you have a key that lets you know what you're looking at. You need to have your data, your X and Y scaled appropriately. Notice I have my temperature degrees Celsius here, so that the X and Y are labeled. You need to make sure that your X is the independent variable, the variable that is not dependent on the other. The Y is a dependent variable that's dependent on whatever you put on X. So the percent females or fraction females depends on the temperature. Then you plot your points following your key, circles for this cross and triangles for this cross. Then you place your errors, plus or minus 8% here, plus or minus 2% here, plus or minus 7% here, plus or minus 3% here, etc. And you do the same thing with this graph. I'm not going to do it. If you did all that, you'd get 100% on that question without and it's just tedious. It's not really conceptually difficult. It makes your life easier if you take, for many reasons, if you take and you go above and below your maximum values. You just choose you have 20 divisions. If you choose 20 different temperatures, then each one is worth 1. Each line is worth 1. So I know that's 21, 22, 23, 24, 25, etc. It's easy. In this one, I know there's 20 divisions and there's 100 total. So then each one is worth 5. So each 2 is 10. So 10, 20, 30, 40, 50, all the way to 100. So just divide it evenly, giving yourself enough space above and below the minimums to be able to graph comfortably. Don't make your graph in a tiny little part of the whole space available. Otherwise, you'd end up with some issues. And B says, construct the graph. I'm not sure why this person wrote this here. They just say B, construct the graph. So it should look something like that, which you would not have been able to do in the AP Classroom. So I'm not sure what they wanted you to do with this question, but I guess this answer is as good as any for what's going on. They describe the overlap between the two error bars in various places. But what they would expect to see is something that looks like this, right? Though in grading these, what they told us is they're looking for, sometimes you'll connect a line, sometimes you do the best fit line, depends on what they're asking for. They're going to be looking for error bars that are relatively the same size, that they're appropriately met. They're not going to be looking at details because you can't put details on here. They're going to be looking that you have a key. They're going to be looking for the appropriate X and Y, data on the X and Y. They're going to be looking for that you have the appropriate title and that you used a good portion of the graph, that kind of stuff. If you did all that, you're going to get 100% on it. And that obviously the lines are pretty near correct. So if you plotted these points, the graph should look something like this. So for each cross, describe whether genetics of temperature determines the sex of the progeny produced. Describe whether genetics or temperature determines the sex of the progeny produced from eggs incubated at 35 degrees. At 35, which would be here, whether genetics or temperature determines the sex of progeny at eggs incubated at 24 degrees. So here. So in order to answer that, let's go back and look at the genetics again. So if it's ZW, it should be female. If it's ZZ, it can be male, but at some point they might turn into females. So as you can see here, that the sex changes depending on the temperature. For that high temperatures, at closer to 25 degrees, 25 degrees, it's going to be genetics. Temperature doesn't seem to be having an impact on, well, we'll see. But in any case, ZW has to be female, ZZ can be male or female. So I guess you have to ask yourself, what do you expect? Well, if I crossed, I'm going to go ahead and do the two crosses, one in yellow and the triangle, which is ZZ male versus ZW female. So if I get Z and Z, and I cross it with a ZW, what I expect to see is, I expect to see at least, if it was just genetics, there would be 50% ZW and 50% ZZ. So that's 50% male or 50% female. That's what I expect to see if it's just genetics. And you see, you get that here. You get that here with the triangle. All the way up until you get around 30 degrees, and all of a sudden you're not getting that 50-50 anymore. They're becoming all female. Because the ZWs are female and the others, ZZ, temperatures impacting them and turning them into female. Okay. So ZW is always female. All right, so let's go back and then I don't remember if they asked me about the other one, but let me go ahead and answer the same question for the ZZ male crossed with a ZZ female. Now here again, if I were going to cross these, I'd expect them all to be ZZ. I hope you see that. 100% ZZ. So if I look at this, it can be male or female. It could be male or female, depending on incubation temperature. So if I'm at lower temperatures, I have 100%. I'd expect them to be at the genetics of it itself. If these are female, these are male. Since I'm getting ZZ, this is going to be determined by temperature. There's no ZW that's going to have any genetics really involved in it, because they all have the same genes. They all have ZZ. They all have Zs. So these chromosomes, there is no genetic difference, right? That we know about. They're all Zs. So the only thing that can impact this cross is temperature. At the lower temperatures, they're pretty much all male. At the higher temperatures, they're all female. So let's take a look at what the question was again. Question C was, for each cross describe whether genetics or temperature at 35 degrees. Well, at 35 degrees, it's up here, it's temperature for both crosses. As you can see, you'd expect to see 50-50. What you're seeing is at 35, you're seeing near 100% females. You'd expect only 50, so that's a big difference. So that's obviously it's temperature that's making a difference. And then what about at 24 degrees? Now at 24 degrees, you'd expect that only temperature is making a difference because all the genes are the same. They're all ZZs. So they'd all either be, so at lower degrees they're all male, at higher degrees they're all female, higher temperatures are all female. And the only thing, there's no difference in the genotype. They're all ZZs. So the only difference is the temperature. So that's with this cross, ZZ versus ZZ. It's a tricky question here, this D, because it says predict the effect of increasing global temperatures on continued presence of W chromosome. The W chromosome is going to be present or not based on whether on the temperature. The W chromosome is present or not whether the parent had the chromosome to give. So if the parent had it to give, the egg or the sperm would get it. There's no data, nowhere in here does it describe the idea that you're going to get a W chromosome or not get a W chromosome depending on the temperature, environmental temperature. So there will be no impact of temperature on whether there is a W chromosome in the population or not. Now the other question, so what could impact, no, that's it. Whether you get the chromosome or not is not really dependent on the environment. There would have to be genetic variability in the population. So you'd have to have individuals that have a W and others that don't have a W. So obviously here you do. You have a ZW or a ZZ. But then you'd also have to have some advantage of having a W or having a Z. So having a ZZ would have to have an advantage at higher temperatures over having a ZW. And there doesn't seem to be any evidence that says that there's some kind of natural advantage for natural selection to take to impact whether someone gets a W or not. You'd have to have an advantage of not having a W. And that is nowhere in here does it say anything about having an advantage over a W. This last D, I'm going to go ahead and say forget it. Just cancel it out. I don't like the claim. The claim to me shows at best it's confusing, at worst it's wrong. So I don't want to waste our time even discussing it. So just go ahead and skip that one. Now, when we look at the second one, it looks like it's easier anyways. Here, going through the steps of meiosis, you're starting with 2 N cells, the next 2 copies of every chromosome, N being the number of chromosomes, 2N meaning diploid, remembering that ploid means chromosome number. Ploid means chromosome number, and 2 is 2 chromosomes, so 2 copies of every chromosome. And 2N meaning 2 copies of every chromosome, N being the number of chromosomes. So, okay, so 2N. So here we start with 2N. And notice there's 2 of these, and there's 2 of those, so 2N. So I don't like that they've drawn this 2N as just a little dot here with a line through it, right? I would rather they actually drew it like so, because that just reminds you the sister chromatids are being separated. But they just, I don't know why they make the decisions they do. I disagree with drawing it this way, but that's fine. You're going to get, according to this, you have half, right, in each of these. But there would be sister chromatids in each of these halves. So you'd have a number 1 here, a number 2 here, and a number 3 here. You'd have a number 1, 2, and 3 here, but there's 2 copies of each of those. And they're not showing those 2 copies. They're not showing you the sister chromatids, but they'd be there. And then the meiosis 2, you divide those. Obviously, here you're doing crossing over and all all the other things we had discussed. All right, so at the end you'd have sperm, right? Spermatozoa. Describe in meiosis that the process in meiosis that ensures both maternal and paternal chromosomes are passed onto each spermatozoan. So this person almost had it right when it says that they meet up. This is what homologous pairs meeting up and exchanging DNA is what guarantees genetic variability. All right. But if you want to know what makes sure that you get the homologous pairs lining up in this way, next to each other, is what guarantees it. And then in meiosis 1, and then in meiosis 1, and then in meiosis 2, what guarantees it is that they line up. The chromosomes line up on the metaphase plate in this orientation, making sure that one copy of each of those gets to each cell. But what determines that you get both maternal and paternal chromosomes are passed on? It's independent assortment, right? So the assortment gets, as they line up, you're not sure in each step in meiosis where those chromosomes are going to end up, but you know that you're going to get one of each of those chromosomes in each of the new sperm. You just don't know if you're going to get this person's mother's chromosome here, this person's father's chromosome there. This is talking about the genetic information. So this is correct. That increases genetic variability. It also makes sure that you get a mix of the DNA. But here they're saying father's chromosomes. Both mother's and father's chromosomes are passed on. So it's this independent assortment of the homologous chromosomes and independent assortment of the chromatids in both first meiosis 1 and then meiosis 2, respectively, is what would be the answer to this question. Explain why genetic content in individual chromosomes and spermatozoa are likely different genetic content in individual chromosomes in a primary spermatocyte. So here at this, here, why would there be genetic, why would this genetic information be different from what's up here? Because of this answer, the homologous chromosomes switching, increasing genetic variability, right? So that's would be, and also that idea of independent assortment. In some circumstances, meiosis of primary spermatocyte and 6 chromosomes will result in 2 spermatozoa containing 4 chromosomes and two spermatozoa that contain 2 chromosomes. Predict the most likely cause. A non-disjunction, so they don't separate. So they don't separate meiosis 2, and you end up with 2 of them in one of these spermatocytes here, and then they split again and they separate, but now you got 2 copies instead of 1 in here of a single chromosome, and they have none over here. A student claims that the animal producing spermatozoa has a mutation in mitochondrial gene. The probability any offspring will inherit a mutation is 0. Student claims that the animal producing a spermatozoa has a mutation in the mitochondrial gene. Why would this? Because mitochondria is inherited from the mother, and sperm is the father. So the only thing the sperm is going to provide is DNA. This is a correct answer. So that's it. This was actually a fairly simple one. It's just complicated because they talk about sperm and they throw in a couple loops with the verbiage and the wording, word use. But other than that, it's a pretty straightforward question. Okay, so when we look at the CD3 and signaling protein that is typically found only in plasma, now see, right away they give you a clue as to as I think when I read this, I see typically found only in plasma membrane of the immune system T lymphocytes. What that tells me right away before I go any further is that I have these cells called T lymphocytes. Let me try to draw this circle a little better. So these things called T lymphocytes they're part of your immune system, as they tell you here. They're the T cells are some of the cells that help destroy things that are invading your body. But that's regardless. That's not important. What it is important is that these are specialized cells, and we've talked about this. They're differentiated. What does that mean? That means that certain genes are turned on and also that of the ones that are turned on, some are upregulated, right? Which means they make more protein, while others make less. That's it, right? So if you have this cell versus, like, say, a brain cell or a skin cell, you'd expect that this cell is gonna make different proteins to different amounts because they have different functions. Each tissue has different functions, so they're gonna need different proteins at different concentrations. So let's look at a muscle cell. A muscle cell is gonna need a lot of contractile tissue, right? So if you have a muscle cell, it's going to have a lot of fibers that are going to let it contract, right? To squeeze together or to relax. So these muscle cells and these muscle cells have to have a lot what are called actin and a lot / myosin. Those are two proteins that are required to really get to have muscles squeeze, contract, and relax. They'll quote me on the spelling. I'm probably misspelling that. But in any case, actin and myosin help our two proteins that work together to contract. You wouldn't expect there to be a lot of actin and myosin in a lymphocyte. There might be some because lymphocytes have to move around, they have to change shape, but I'd expect there to be more here. So this gene would be upregulated. In other words, you'd have to have a way to turn up the production of. So already with just the first sentence, I'm thinking about all this. So that's what I have. And then is composed of different polypeptides, including a gamma chain CD3, and this little symbol is a Greek letter gamma. So CD3 gamma is a protein. Scientists analyzed the promoter of CD3, a gamma gene, for regulatory sequences that might have positive or negative effects on expression. So they looked at a gene. So if we have DNA, If we have, if we have DNA, and in that DNA there's a gene, for this CD3 gamma that's found in, I can't really do a gamma, but I'll do something like it. CD3 gamma gene that's going to be found in T lymphocytes. And there's a region, some part of this early part of the DNA before you get to the gene is regulating it. So this is the promoter region. The promoter region allows for the gene to be expressed. It says, yo, this is what we need this gene expressed. And remember how promoters work. You have these specialized transcription factors. So I'm just going to put circles and squares and triangles. But there are these factors, they're called transcription factors, that are going to bind to the promoter region, that are going to allow for the gene to be turned on. These transcription factors are made specifically for the cell type, for that tissue. So if you have a brain cell or a muscle cell, you would not expect that there would be transcription factors for this gene if this gene is generally found in a lymphocyte. Right? Because you'd expect that it's this protein specifically for a lymphocyte, or at least it's going to produce more of this protein in a lymphocyte than it would somewhere else. But let's go ahead and read on. I don't know what the question's actually asking. That's just the stuff that I know. Always when I read a problem, I try to read what they're telling me and then remember the things that I know. This is stuff we've covered in class that I know because my teacher taught it to me. So I try to make sure I kind of take notes as I go along. Including gamma chain, scientists analyze the promoter region and for regulatory sequences that might have positive or negative effects. So they're telling you in here, they're asking, there might be portions that downregulate it. And there might be portions that upregulate it or inhibit, right? So downregulate would be inhibit, or turn the amount of protein being made down, amount of mRNA made, and therefore the amount of protein down. Or it could be promote or encourage. So promoter regions generally start the process, but there are nuances, right? So you control it very fine, like a dimmer switch. You can inhibit or turn it up or down depending on what you need. So that promoter region, that region before the gene, is used to regulate the gene production. It can be pretty far up when it comes upstream. But I should tell you, and we talked about it in class, but if you're, let me change the color. If you're reading this in this direction and making mRNA, if this RNA polymerase is reading this DNA and making mRNA as it reads it, then this part down here, we call it downstream. And up here, we call it upstream. Just like a stream flowing water, downstream means in the direction of the water moving, towards the direction of the water moving. Upstream is in the opposite direction. So upstream versus downstream. So that's all the stuff I know from reading and from class. All right, moving on. So, there's the word upstream, so I'm glad I talked about it. Scientists cloned fragments of CD3 gene that included the first transcribed nucleotides plus 700 nucleotides upstream, 789. So, upstream. So, upstream is... Oh, I shouldn't even do that because that should. This part, 789 nucleotides. And they also added to their clone about a few of the actual nucleotides that are part of the gene. So some of the yellow. And 789 nucleotides before the gene. So you do have some of the gene plus that being cloned. What do we might clone? We take that sequence and we put it into a vector. What's a vector? It could be a yeast. It could be a bacteria. It could be a combination of that. It could be a cell. It could be just a regular human cell. But we take that and we let the cell make copies of that DNA for us. That's what we mean by clone. When we mean clone, we mean we make copies. Equals making copies. All right. That's a lot of ideas all in a few sentences. This is what makes AP tests hard. 789 nucleotides upstream sequenced into plasmids in which the gene for a firefly enzyme luciferase immediately follows the fragments. So let me see what else they say. Let me just read the whole thing and then I'll go back and explain it. The plasmids are then introduced in a line of T lymphocytes. The cells were allowed to grow, and that's how you clone it. You put the gene in the plasmid, and then you put the plasmid into the T lymphocyte and allow the T lymphocyte to do mitosis. That copies your DNA. The cells were allowed to grow in a short while because the regulatory sequence of CD3 immediately preceded the luciferase of the plasmids. The activity, either positive or negative, the regulatory sequence affected the amount of luciferase gene expression by the T lymphocyte. Luciferase catalyzes the reaction that results in the release of light and is responsible for bioluminescence, light flashes of fireflies. By quantifying the bioluminescence luciferase, the scientists were able to determine the effects of CD3 gene fragment cloned into the plasmids on the expression of the gene. All right. I don't know why they have to make it this complicated, but fine. So they took into that T lymphocyte. Remember, a T lymphocyte has a nucleus, is a Golgi body. It's a eukaryote, right? It's part of eukaryotic organisms. Has all that. If you take this vector, in this case, they're not even using a vector. The vector is the T lymphocyte. They're going to make this a plasmid, which is a round piece of DNA. It's closed off. They're going to add the promoter region, the 789 nucleotides of DNA that comes right before the gene. They're going to add a little bit of the gene. And then, because what they're interested in is looking at this promoter region and asking, is it down regulating it? Is it up regulating it in the T lymphocyte? They don't know. So they put this regulatory sequence in. And then they probably want to know what part of it is down regulating it, what part of it is up regulating it. So they put it all in there. And then they add a little bit of the actual gene. So it crosses over. So they make sure they get the whole promoter region. So we know it's going to express. So this is what's going to tell you, make the gene. But if you just made the CD3 gene, you'd have to somehow have a way to isolate the CD3 gamma from everything else. But luckily, one of the things we can do is add another gene instead of the gene that we're looking at. And that gene could be a protein that glows for us. So this gene, this is the gene for luciferase. Double check that is it, yeah. So remember, what does that mean is that if I have a lot of luciferase, it's going to glow. If I don't have a lot of luciferase, it's not going to glow as much. So the more luciferase I make, the brighter the glow. It's bioluminescence. You know that. You've seen fireflies, I'm assuming. In fact, I'm pretty sure you have. And that's it. That's what they're going to do. And they're going to, let's see what they do with it. But so again, they put that construct into a T lymphocyte. The T lymphocyte does mitosis. It expresses the gene. Why is it going to express the gene? Because it has the promoter region that in a T lymphocyte, you're going to find the transcription factors in the nucleus that are going to bind to the regulatory sequence and turn this luciferase on. And it's going to make a lot of DNA, or a lot of protein. And the protein in this case is an enzyme that glows in the dark and makes the organism glow in the dark. All right. Bioluminescence is what it's called. It's one of the really important techniques in modern molecular biology. Allows us to identify whether a gene's working or not. So as here they have a quick drawing of an upstream regulatory sequence. The +1 means that's where it starts to transcribe, right? The DNA is going to start to get turned into mRNA. So they take that part and they put it in the plasmid. It's a round DNA. And they have the location in which the CD3 sequences are inserted. So this is where you're going to insert it. Here you have the luciferase gene. So this part is put in right here. Then right after it, there's the luciferase gene. So if this is upregulating it, I'm going to get a lot of luciferase. So it's going to glow really bright. If this is downregulating it or doesn't or what parts of this upregulate or downregulate, it depends on the promoter region. So if everything's working right and I put it into a T lymphocyte, it has to be put in a T lymphocyte because the transcription factors for this gene, which comes from a T lymphocyte, are found in T lymphocytes. And it may be very different in a muscle cell or a brain cell or what have you. All right. So that's kind of a summary. I love when they give you this kind of summary of what's going on. They put it into the plasmid, into the T lymphocytes, and then T lymphocytes with plasmids make the luciferase enzyme, which makes the cells glow in the dark. And with a light meter, you can measure how bright it is. So let's take a look at this. All right, it looks like what they did, and let me read real quick. Yeah, that's exactly what they did. So they took and they graphed the amount of luciferase activity against different portions of the promoter region. So in here, these two don't have, this has the parent plasmid, just luciferase, with very little of the promoter region there You get only a little bit of light. Here's a parent plasmid with non-CD3 active promoter. So this is the promoter, a promoter region, and the parent plasmid with an active promoter and the luciferase enzyme making a lot of light, right? And here it is, the parent plasmid by itself. Parent plasmid by itself without the non-CD3 active promoter. So I'm gonna, because of how much luciferase, how much is being produced, I'm gonna go ahead and assume that this is just a general promoter here. The core promoter is being added to this. They don't mention it, but you're making so much luciferase activity, you're making so much luciferase, it goes way beyond anything else, that this must be a core promoter region that's definitely a positive control. So here you have something that you're making a lot of it, and for sure it's doing its job. And this is how much luciferase is made without, just the plasmid without the promoter, right? This is with the non-CD3 active promoter, so this is an active promoter that just turns it on, 100% on. This is without a promoter at all. This is what it looks like, how much luciferase you'd make if you didn't have any promoter at all. And then this is a combination of the promoter regions, smallest to largest, right, upstream. So here's 15. There's a little segment of the 15 nucleotides of the promoter region. Here's 59, and I'll draw it out for you. Let me just draw it out right here. So if I were looking at this, you'd have a little bit of the promoter region, right, 15. And then you have the luciferase enzyme gene. So you're making a lot. So I would say this little bit here, the first 15 nucleotides, you have definitely positive up regulation, so you don't have a down regulatory impact at all. Where when I get to 59, somewhere in there must be some part of that promoter region that's between 15 and 59 that's down regulating it, because you see it, you have here about 10 on the luciferase scale, activity scale, where the next one you're down to less than 2, right? Less than 2 here, and then the rest. This is the least of this section. So the negative regulation sequence is definitely happening here. So there's some positive influences between 59 and 123. You can see it going up. At 199, there must be a little more positive, but this negative is still impacting it because you're not as high as the first 15. You're not as intense. There's not as much luciferase being made as the first 15 nucleotides. So when I look at this chart and I look at this, and by the way, this is the CD3 gamma, the plasmid, the CD3 gamma, 15 nucleotides upstream, 59, 123, 149, all the way up to 789. All right. The first question, and these are answers from someone in the room, in your class. Identify both cellular component and the location of the component that is responsible for producing luciferase protein from mRNA described in the plasmid containing T lymphocytes. That's easy. That's the ribosome. So these guys got it correct. It's the ribosomes. Explain what dictates to the lymphocyte the correct order of the amino acids should be. Well, that should be mRNA. And somebody here got it right. It's mRNA. So that's easy. Basic transcription translation. Not a big deal. Easy peasy, good number, a good portion of FRQ already done. Identify the independent variable in the... Now, I just want to point out, that was all very complicated. I think very doable if you broke it down, but it's all very complicated, as you can see. But the first question in the FRQ is really pretty easy. It's what is like ribosomes and mRNA. Pretty straightforward question. Straightforward answer. Identify the independent variable in the experiment described. The independent variable, there's two things that are changing, right? You have how much luciferase you're making, how bright the light is, and then you have how much of the regulatory sequence you added. So how much regulatory sequence you add is the independent variable, and how much light is produced or how much activity there is the dependent variable. So the more or less you have it, that changes how much luciferase is being made. All right. So let's see, independent variable is plasmid introduced to lymphocytes. That's true. How much the plasmid itself is the independent variable, or how much of the regulatory sequence is added is the independent variable. And identify the plasmid used as a negative control. Well, that would be the parent plasmid. And justify including the plasmid with non-CD3 active promoter. That is your positive control. Well, this person got it perfectly right. I hope they understood it. I hope everything good. It's so right. In fact, and that's perfectly stated and well executed. It's actually directly from the grading rubric. So I'm hoping that they didn't that they actually wrote that themselves. But I'm moving on. I'll talk to them individually. But I think that's so close to the actual rubric answer. I have concerns. All right. Identify the plasmid that must contain CD3 core promoter sequence, but the fewest or no negative regulatory sequences. Identify the plasmid that must contain CD3 promoter sequence, but the fewest or no negative consequences, regulatory sequences. So in the promoter region, you have to have a promoter region, so they have to have lit up. They have to be lit up, and which has the least number of negative regulatory sequences. Well, it has to be this one, right? The 15. I'm trying to find where that was. Wow, I just keep reading the student's answer, and it just justify including the plasmid with non-CD3 gamma active promoter, non-CD3. All right. The negative control was the parent plasmid. The CD3, the non-CD3 was a positive control for good to new plasmids. Okay, I mean, good intro to new plasmids. I'm not sure what they meant by that. I just keep reading that, but that encourages me that they're writing their own answer, because I'm not sure what they meant by that. But I'm going to move on. So. Again, identify plasmids that must contain the promoter region. What we just said has to be that 15, right? And describe the most likely cause of variation in luciferase activity among the cells that contain one. Why are there variations in how much luciferase is being made? None of it. All right. So I'm not sure what they meant by that, but let's put that out of the way. And let's get rid of this because I'm not sure what they meant by that. So I'm just going to go ahead and write the answer here. Identify the plasmid that must contain CD3 gamma core sequence, but the fewest or no negative regulatory sequences. That has to be the plasmid that's CD3 gamma 15, right? Because it has the most luciferase being made. And there it is. CD3 gamma 15 has the most, right? Not outside of the experimental groups. That's the one that has the largest, right? you might and we already said that this one here, so this one here, the positive control of the CD3, the fewest number, no negatory, C315 of the experimental group is what I would write. But honestly, if you wanted to write down an absolute perfect answer, you'd have to write down the parent plasmid well, it says non-CD3 gamma, so I'm going to go ahead and not include that one because it says non-CD3 gamma, and this says contain the CD3 core promoter, so this doesn't have the CD3 core promoter, so it is the 15, CD3 gamma 15. I was kind of debating how to phrase that, but this is obviously the answer. Based on the question, the data in Figure 2, describe the most likely cause of the variation in luciferase activity among cells that contain 419, 309, 239, and 199. Right? What's the cause of having those differences? So if I look at the differences we just talked about a minute ago, this is producing a lot more of the luciferase. This is producing a lot less. This is producing more. This is producing less. And I think you see 419 is a little less. 309 is more, etc. So what would explain the differences in that? And the easy answer is the various percent regulatory sequences versus down regulatory. So some of those sections are going to be upregulated, some are downregulated, and depending on, so for instance, if I have, and you can phrase it any way you want, if I have this one upregulate, this part of this, so here the regulatory sequence, maybe this is upregulating, they're being upregulated, but then, so I get, that's like 15 in, so that really gives me a lot. Let me just do it this side. This is 15 in, so it gives me a lot of luciferase. That would explain the CD3 gamma 15. But then as I move on and I have this section here might be downregulated, then I might have another section might be a little more upregulation. So it encourages, this discourages the production, and maybe this one encourages some more. And so depending on whether I have just this one by itself, or these two together, or these four together, I would have parts of it that are going to encourage more luciferase, other parts encourage less. So depending on how much of the positive or negative regulation I have in that promoter region, I'm going to get different amounts of luciferase being produced. Calculate the percent increase in luciferase activity between cells containing CD359 here, which is about less than 2, a little less than 2. Let's just say 2. It's not 2, but let's say 2. And 149. 149 is about 4. So from 2 to 4, that's 100%, right? Which is actually somebody, they actually wrote 100%. So I'm not sure how they got that if they didn't understand the first part of it, but maybe they did and I just didn't understand what they wrote. So the last part of that answer, so if this is I, this is II. The third III of 3 would be 100%. So going from 1 to 2, or doubling it. right? It's a 100% increase. All right, going from 1 to 2 is a 100% increase. Last question is predict how likely observed levels of luciferase activity in CD379 is introduced to non-lymphoid cells, such as a kidney cell, to provide how much luciferase would we get? What's the level of luciferase? Very little. So I would predict very little to none. No luciferase. And why would I? That's my prediction. Why would I predict that? Because kidney cells have different transcription factors than T lymphocyte cells. So the gene would be less transcribed if at all, right? And that takes care of that question. It's a complicated question, but doable. And this person showed that they could do it. They had one question that I would have marked at least partially wrong. C, I would have marked part of that question wrong. Like probably the first I, like little I and little I would have marked wrong. They have 100%, but the way they phrased it, the percent increase would be 100%. That would have been right. I would have said right, correct on that. They didn't ask you to show your work or anything like that. And here they said justify. I probably would have given them 2/3 of this question, this part of the question right as well. Probably got I would have said I and I would have been right, but the last part, the triple I would have been wrong. The justify part would have been wrong. It just doesn't make sense. But everything else would have been right. So you would have had majority of the question correct. So that's pretty good. The DNA replication question I'm going to leave till Thursday. I want to go over DNA replication with you, and then maybe just do this actual question with you in class. So if you did it, great. If you didn't, don't worry about it. We're going to go over that in class. All right, that's it then.